Sorry for that. I needed it.
So I'm going to discuss our current thinking on how the EGF receptor switches on when it receives ligand. And part of the reason this issue became interesting to us was because the classical model for how receptors activate, which is to phosphorylate the activation loop, so that when the kinase dimerizes upon ligand arrival, the activation loop phosphorylates and switches on activity. This idea, introduced first by Yossi Schlesinger, doesn't actually apply strictly to the EGF receptor. And the reason is that the activation loops or activation segments of the kinase domain of the EGF receptor, although it has the tyrosine residue, doesn't actually require phosphorylation for activity. And this was first shown biochemically and also by a structure that was determined at Genentech, in which the activation loop is not phosphorylated, but the kinase remains active. And we and others have shown that if you mutate the tyrosine phenylalanine, the receptor will in fact activate properly. So the mechanism of activation cannot rely on this receptor transphosphorylation. However, there must be a control element in the kinase domain because, as you may know, the EGF receptor phosphorylates itself on a long tail, which is about 200 residues long, longer in terms of in some of the family members. And if you look at the sequences of these tails, they have characteristic features of unstructured elements. And so the kinase did not have a control mechanism that kept it off, all hell would break loose. And so it must be controlled. The question just is, how is it? And so the clue the first clue that we got was from a structure determined at Glaxo by Wood et al. Louise Johnson has already shown this structure, and we recognized that, and the authors recognized and pointed out in their paper, that the structure of the kinase domain that bound to lapatinib is that of inactive SRC or inactive CDK. It's a very characteristic conformation here, but you might wonder whether that inactive conformation has bearing. I noticed I'm not wearing this microphone. Is it still picking up?
Yeah.
Okay. So you might have wondered whether this has any bearing on the actual mechanism, except for the fact that a very large-scale biopsy study carried out in Japan on lung cancer patients who had EGF receptor mutations made the remarkable observation that 40% of the patients had a mutation in a single leucine residue, this one. And when we saw this, we immediately realized that this is pointing to the importance of the inactive CDK structure, because in the active conformation, the leucine appears to be in an innocuous position, whereas in this inactive CDK structure, it's like structure, it's part of the hydrophobic interactions that stabilize the inactive state. So the idea is that the kinase, when mutated at that site, and so Shu Zhang, who did this part of the work with me, showed that if you introduce a single mutation into the kinase domain at that leucine, you activate the kinase, so undoubtedly it must be inactive. And a couple of years ago, we published a paper in which we reasoned that the fact that we always see, Genentech originally, but my group and Michael X group always sees active EGF receptor, is because there's an interaction in the crystal structure, which we identified, although people hadn't really noted it before. And what happens is that in the crystals, we see a dimer of EGF receptors in which one EGF receptor molecule plays the role of a cyclin. And it doesn't look like a cyclin, but there are a number of analogies between the interaction between the yellow kinase domain, which we call the activator, and the blue kinase domain, which we call the receiver, whereby the receiver is switched on. And just to give you one glimpse of this from our published work before I move to the new work, if you look at the interface between the kinase domains, the receiver is in blue and the cyclin-like activator is in yellow. They form a hydrophobic interaction. First, there's a pocket on the receiver kinase domain, which is filled in by residues from the activator. I'm not going to show you all our experimental data that are published, but if you make point mutations at this interface, you kill signaling. And one way to look at it is that as the kinase switches from the inactive to the active conformation, it opens this hydrophobic pocket, which is next to helix alpha C, and it requires, it appears, filling in of this hydrophobic pocket by the activator. And as I said, I'm not going to cover the old experimental data that's been published, except to show you a cute result, which is if you make a single mutation, which is a valine to arginine mutation at the base of the C lobe, and now crystallize the kinase domain, you no longer get the Genentech structure, so to speak, because it can't form. Instead, what you get is the inactive CDK structure. Now, note that the mutation is at the very base of the kinase domain, very far from the active side. But here it is in the CDK conformation. And if I now bring in lapatinib from the Glaxo structure, you see that lapatinib, in fact, induces no structural change. It's just selecting the structure. And Dan Leahy and colleagues have shown that HER4 forms the same inactive structure and the active structure, so we think this is a general mechanism. And one more thing I'll just point to is that Shulu Zhang, before he left my lab, determined the structure of an inhibitor of EGFR called MIG6, and that works by blocking this activating surface. And all this work has been in collaboration with Phil Cole. I forgot to say that, but his stern visage right in front of me reminded me to say that without Phil, we wouldn't have been able to do this. So what I want to focus on today is the mechanism whereby the EGF molecule signals to the kinase domain. And in the interest of time, I won't describe the extensive work done by many groups on the structures of the extracellular domains, the work of Burgess and colleagues in Australia, Kuriyan and colleagues in Japan, Dan Leahy, and people at Johns Hopkins, and Mark Lemon at the University of Pennsylvania. So our view of how the EGF receptor activation process proceeds is actually changing from the work of, I've mentioned some work from Burgess and another group here, but essentially a simple monomer to dimer equilibrium is unlikely to underlie activation because there's a lot of evidence for preformed inactive dimers. And particularly Burgess and colleagues using fluorescence have pointed out that in fact there may be higher order structures such as tetramers that underlie formation. So this is kind of the background. Now, with this background in mind, we were gratified when, in our tinkering with the kinase domain of EGFR, Natalia Jura obtained an inactive dimeric structure of the EGFR kinase domain. Like any of these structures, you have to ask whether it's relevant, but I'll point out several things here and show you aspects of the structure that tell us that it's relevant for maintaining the inactive state. The C-terminal region, which is the first portion of the substrate phosphorylation sites in this kinase, are tucked in symmetrically here. And you'll see that the C-terminus has a helix, which we'll call the AP2 helix, for reasons that have become clear, that packs against the C helix of the adjacent kinase domain. And then there's a turn, and you'll see that the C-terminus runs along the surface of the kinase domain. So here's the structure on which that's based. And so now if you look at it a little bit in this closer in this animation, what you find is that the C-terminus forms the alpha helix and removes the other kinase domain and then makes a turn and comes down. And so this turn is positioned near a site which we're very familiar with in the kinase field. It's the back of the hinge region, the alpha C beta 4 turn, and the hinge region of the kinase. And there's a network of ionic interactions which resemble inactivating interactions that we've identified in ZAP70, where different structural element forms salt bridges like this. And Musa Mohammadi has identified that these kinds of salt bridging interactions can turn off other tyrosine kinases. So we see something we recognize, and it's also propagated to the other molecule in the dimer. And so if you now go in and mutate these residues, and so what's done here is to show you 3 mutations. 2 of these are double mutations in the acidic residues, and one of them is in, if you like, a receiver lysine residue. And what you want to note here is that when you introduce these mutations in the C-terminal tail or in the kinase near the hinge region, the kinase essentially becomes EGF independent. So that's a very significant result, we think, and points to the fact that this electrostatic hook, as we call it, is probably doing something to keep the kinase inactive. Now we turn to this alpha helix that presents the electrostatic hook. And when you go in and rotate in and look at this alpha helix, now Frank Zakary worked with me on the structure of inactive Src kinases, and he might recognize it, if no one else does, that this is actually sitting almost precisely where the SH2 kinase linker sits in an inactive Src kinase. And if you zoom in, you find that this alpha helix is presenting residues, hydrophobic residues, much like the SH2 kinase linker does. And so, I have to tell Natalia to speed up this movie a little bit. You'll see that it presents, for example, a phenylalanine residue, and in SARC, this is an isoleucine. And when you take the isoleucine, you'll activate SARC. And so the C helix is over here. So this looks good. And the other interesting thing is that this alpha helix actually contains the plethrin adapter protein AP2 recognition motif, which will take EGF into endocytic vesicles. It's one of the mechanisms. And it's also known that the AP2 recognition motif of EGF receptor is hidden until EGF arrives. So that's an intriguing aspect of the structure. Now, if you mutate this AP2 helix, which has the SARC-like role, you get an activation in our in vitro assays, suggesting that there's an equilibrium in vitro on vesicles between dimeric and monomeric forms of the kinase. But the surprise is that when you mutate this AP2 helix in cell-based assays, where you're measuring phosphorylation of the tail of the receptor, you don't see activation. In other words, if this were an autoinhibitory element like the electrostatic hook, when we mutated the electrostatic hook, we saw activation. And so if you mutate an autoinhibitory element like the SARC SH2 kinase tail, you should see activation. Instead, what we see is inhibition, and I'm going to return to this. So now I want to turn, so we have a candidate for an inactive dimeric cytoplasmic portion of the EGF receptor. And I want to return to the activation mechanism. And so schematically, what the EGF receptor looks like is it has an extracellular domain, transmembrane segment, a juxtamembrane segment, and then a kinase domain. And we're going to focus our attention on the juxtamembrane segment, which is highlighted here. It's about 40 residues, roughly. Now, what Carpenter and Thiel did in work that was published earlier this year is to make a construct of a kinase domain that includes the juxtamembrane segment. So it's the entire cytoplasmic portion, and they transfected cells with these constructs and then looked at phosphorylation of the EGF receptor. And what they found is that if you add the juxtamembrane segment, which I indicate here schematically, strongly activates the kinase. And so here, for example, if you have an impact juxtamembrane segment and you look at phosphorylation on the receptor is switched on. And if you cut off half the juxtamembrane segment, you lose it. And if you cut off all the juxtamembrane segment, you're essentially dead. And so this means that the juxtamembrane segment in EGF receptor is doing something very different from mechanisms that operate in receptors such as EphR receptors, which Frank Zakary defined, or in FLT3 or c-KIT, where they are autoinhibitory segments. Here, deleting the juxtamembrane segment releases an activation process. So what we did, because the cell-based assays might involve factors other than the kinase alone, what Natalia did was to purify different constructs of the kinase and then measure activity against the synthetic peptide based on the EGF receptor tail, one of the phosphorylation sites. And a remarkable result was obtained, completely consistent with the results of Carpenter, but nevertheless to us remarkable, which is we get a 70-fold increase in the specificity constant for catalytic efficiency, kcat over km, by simply adding the juxtamembrane segment. And if you delete, this will become important for the analysis that follows, if you delete half the juxtamembrane segment, you lose 10-fold activity. By using complementation, where we make a mutation, the valine 924R mutation that I showed you earlier, a crystal structure of, that mutation blocks asymmetric dimer formation. So if you introduce this mutation, as Carpenter showed in his cell transfection studies, you kill this effect. So in other words, the asymmetric dimer underlies this activation of the EGF receptor. Likewise, if you make a mutation in the N-terminal lobe of the kinase, which serves as the receptor or receiver, you kill this effect. If you mix the two of them together, these half-impaired kinases, you restore activity at a level that's probably statistically reasonable for kinases where only one partner can work. So it looks like what's happening is that a dimer is forming that's like the asymmetric dimer. And then you'll note that if you have only one of these juxtamembrane segments, you lose activity. So it looks like you need both juxtamembrane segments. And the juxtamembrane segment basically dimerizes the kinase domain. If you do light scattering, which has to be done, as you may know, at high concentrations, the kinase core is a monomer. But if you add the juxtamembrane segment, it becomes a dimer. I'm not showing you those data. What I'm showing you here is that if you change the concentration of the kinase, and again, we're limited because the substrate is a poor one, kinase activity is weak, our assay only lets us realistically go from 0.5 to about 10 micromolar we don't see a change in the activity. So that means that the kinase is a dimer most likely stronger than 1 micromolar dissociation constant. So in order to, again, test how good this is as a dimerization, what we did is to make the following constructs. So the first construct we made is to include the transmembrane domain, the juxtamembrane domain, and the kinase domain, but no extracellular domain. Now, to compare, so we're going to get an activity, and it's known from the work of others that if you remove the extracellular domains, you activate the kinase. So we expect this to be active, and it is. So that in order to give us an idea of just how active it is, how good is it, what we did is to fuse GCN4 to the juxtamembrane kinase domain to form this construct. Now, GCN4, as you may know, is a constitutive dimerizer. And then we transfected cells with these constructs and looked at the activity. And what we find is that, let's look at the transmembrane juxtamembrane kinase domain. It actually switches on. So this is a cell transfection experiment. These are protein levels. This is phosphorylation. Now, if you delete the transmembrane segment, and under these levels of transfection you don't see activity, we take this to simply mean that the concentration of the kinase is lower than the dissociation constant for the juxtamembrane. What happens is when you take this and put it on the receptor, you increase the local concentration and the transmembrane domain can do something. But without it, it's at low concentration in the cells. Now here's the GCN4 construct. You'll see it switches on. And the activity is essentially the same as that of the TM construct. If you make the valine mutation, you kill it. And that's an important result because you know that any kinase can be activated in principle by simply co-localizing it, allowing transphosphorylation. But this is actually a specific interaction involving asymmetric units. So the bottom line is, if you remove the extracellular domains, the kinase is essentially constitutively dimerized. That's our interpretation of this. So another question becomes, how does the juxtamembrane segment do this? So if you look at the sequence of the juxtamembrane segment, it has 3 portions that are important, or 4 portions. There's a portion that's already present in our crystal structures, and we understand what this does, and I'll show you. Then there's a region here where there's some acidic residues. Then there's a LXXLL motif, where X in this case is arginine, and then there's some basic residues. And so we're going to analyze this in turn, but first what I'm going to tell you is that Glaxo published a structure of inactive HER4. And they didn't comment on this, but it turns out that they introduced this juxtamembrane, this portion of the juxtamembrane segment. And by looking at that, we gained considerable insight from stuff that's in the protein data bank. So simply put, what they did is they put in a covalently bound inhibitor on the right-hand side in HER4, which actually causes the kinase to be in an inactive conformation. So this is analogous to Luis's description of the CDK9, Luis, cyclin T, because what's happened is that the activated kinase is actually there. The activator kinase is actually there, but it's partially disengaged. And the receiver kinase is in an inactive conformation. And that's food for thought, but I really just want to show you that the juxtamembrane segment is begun to be seen. So here's what's seen in the HER4 structure. And if you look at how this juxtamembrane segment interacts, you'll see that it runs along a track here which involves conserved residues that are conserved in EGF receptor family members. So it looks like it's something interesting. And in fact, if you go through this set of interactions here defined in the HER4 structure and mutate these residues, it's very strong and very consistent. You'll interfere with EGF receptor activation. So this looks like it's real. And one of the things that's also interesting is that the MYC6 inhibitor protein that we had defined earlier, which binds down here, has a segment that has the same sequence as this. So we're pretty sure that this is what the juxtamembrane region, or at least a portion of it that we call juxtamembrane B, is doing. It's providing a latch, and we'll call this the juxtamembrane latch, that allows the receiver kinase to hook onto the active kinase.
In that picture, can you just say where you don't see this? Is it under your surface?
This must be here, C. I imagine that's here, C, yeah. Yeah. Anyway, we have this juxtamembrane latch. Well, how did I completely lose my place?
Right back.
Going backwards.
Where am I? So the other startling thing was that if you remember, our inactive kinase dimer had this AP2 helix, the electrostatic hook, and then a strand running along the surface of the kinase domain. It turns out that the strand that the C-terminal region now in the inactive structure presents to the surface of the kinase domain runs along the same track that the juxtamembrane segment is using. So to summarize this here, we have an inactive dimer in which the C-terminal helix runs end to C-terminus in this way. And we have an activated dimer based on the Glaxo-Herford structure in which the C-terminus, the N-terminal juxtamembrane segment runs end to C in this direction. Near perfect mimicry. So now what happens? So we look then at the rest of the juxtamembrane segment, and there is a pattern here of leucine at the A position, leucine at the D position, leucine at the E position. There's also a pattern of arginine and glutamate residues that are spaced 4 apart, and so it's very reasonable to assume that you have 2 alpha helices. And if you put them in an anti-parallel orientation with standard geometry, you'll find that the first leucine, which is on the left here, packs in a V-shaped crevice formed by the other 2 leucines. And if you tinker with these leucines, you kill EGF or severely impair the responsiveness of the kinase to EGF. Now, what's particularly nice is if you just build a short anti-parallel alpha helical structure as shown here, these arginines and glutamates that I have indicated to you end up forming a network of ion-pairing interactions. And in the interest of time, I'm not going to show you this, but what we did is to run about 5 or 6 molecular dynamic simulations, each extending about 10 nanoseconds, and we verified that this is, it doesn't prove the structure is right, but it at least verifies that this is a reasonable energetic model because it stays stable and the ion pairs flicker in and out, but basically stay good. So we think that what's happening then is that the end of the juxtamembrane segment forms an anti-parallel coil-coil or dimer interaction, which can be simply docked onto the crystal structure provided by the Glaxo group. But then something remarkable happens in a stream of remarkable observations. Remarkable simply because I wasn't expecting this. If you look at the transmembrane segment of the EGF receptors, there are these dimerization motifs. And very recently, from a group in Russia, we have the NMR structure of the transmembrane segment of ERB2 done in a lipid bilayer. And it's actually as expected from modeling, so in one sense there are no surprises here. But there's this characteristic pattern of the splayed apart helices dimerized using one of the glycine motifs as people like Mark Lennon and Mark Sternberg Sternberg had predicted years ago. But what now happens is if you look at the distance between the ends of the helix, which is the beginning of the juxtamembrane segment, in this experimental structure, it's 20 angstroms. And if you look at our anti-parallel coil-coil, which is based on the leucine patterns, that's about 18 angstroms long. So it's obvious these things will just fit against each other, leading us to conclude that what happens is that the transmembrane segments can form this structure which stabilizes the asymmetric dimer. And that probably the job of the extracellular domain is to prevent, in the absence of ligand, close acquisition of the transmembrane helices. And that we know from experiments other people have done that if you put in a flexible linker between the transmembrane segment and the extracellular domain, you'll activate EGF receptor. So that leads to this idea that in the absence of EGF, you have inactive monomers that, and we think they might form inactive dimers like this, but the active dimer is destabilized in some way because the helices can't get close together. And remember that the juxtamembrane A segment, which forms the alpha helices, are essential in our experiments for the dimerization. So if you hold them apart, the prediction is the dimerization will be impaired. And that what EGF does, and we know from crystal structures of the extracellular portion, is bring the tips of the extracellular domain close together, which will at the very least facilitate and possibly even promote formation of the active dimer. I'll just end with an observation that another interesting observation we made. So if you go back to this structure, this may be all that's required. If the tails are long, you've activated one of the kinases, both tails can enter the kinase domain and be phosphorylated, and that's the end of the story. But we noticed something very interesting. I'm not going to take you through sequence analysis, but if you analyze the sequences, you realize that there are 4 EGF family members, EGF receptor family members, of which one, HER3, is kinase dead. So the idea would be HER3 only takes the activator position because it has no kinase activity. But it turns out that in HER3, every element I've described to you of this juxtamembrane latch on the receiver side is preserved. So it looks as if nature wants to put HER3 on the receiver side, and we ask, why might nature want to do that? And one thing that occurred to us is that here's Susan's PKI, the protein kinase I substrate mimic, and I think that's how things are going to enter the active site in every kinase, including EGF receptor. And the C-terminal tail of these kinases, the EGF receptor kinases, is the interactions involving the AP2 helix, among other things, that keep it going off in the wrong direction. And so remember, the AP2 helix killed the kinase when mutated. Now, it's just a simple geometrical fact that if you extend the asymmetric dimer, so here's an asymmetric dimer up here, activator, receiver, and if you just bring in another receiver to take a third position, it turns out, I'm going to do this for the inactive dimer. So here's the activated kinase. This is switched on. Here's Susan's substrate to tell you where the active site is. Here's our inactive monomer. And you'll see the C-terminus actually points in the right direction even though it turns around. In this case, if I build a dimer, I'm going to now build an inactive dimer. What happens is in the inactive dimer, the C-terminus is positioned exactly right to enter the active site. And if it were a monomer, it would just have to turn around and come back in. And so I'll end here by suggesting that in addition to activation, there may be mechanisms that use the receiver interaction in an asymmetric dimer. So this kinase here is inactive, technically, but it's in a receiver position. And there's some geometrical arguments that suggest that this might actually favor transphosphorylation. This is an interesting idea for which we have no verification. So Natalia Jura is a postdoc currently in my lab who did much of the work, the new work described here with Nick Endres and Kate Angolds and help from Sebastian Dingel. And Shubu Zhang discovered the importance of the asymmetric dimer when he was in my lab. And I've already acknowledged, Phil, I've also mentioned people at UCSF that we're working with and partnering. Thank you.
John, so the EGF receptor also can dimerize with other tyrosine kinase receptors.
Well, with EGF receptors.
Well, with the PGF receptor as well.
I'm not aware of that, so those details
well, I was just kind of wondering whether you...
So I was completely, I mean, to be honest, I didn't know that there was any physiologically important dimerization with any. We've analyzed EGF receptor family members in great detail and have residue by residue maps of how they might interact. But
yeah, it might be.
EGF,
Yeah,
you
might
can tell
be,
me afterwards
yeah.
what you're referring to. Phil.
So, what do you think the role of cetuximab, which is an antibody that can inactivate EGFR, is to prevent dimerization or to keep it in a symmetric dimer model?
Oh, I don't know if it keeps it in a symmetric inactive dimer. My current thinking, based on everything I've read, is that at least at a reasonable level of approximation, what you want to do is just prevent an intrinsic activation ability in the kinase and transmembrane segments. So anything that just prevents them from getting close together. So they may even be in contact, but as long as the geometry is such that the juxtamembrane helices can't form, then that's our current thinking. And it's consistent with a lot of what's in the literature.
Did I hear correctly that when you included the juxtamembrane region, the protein, along with the kinase domain, the protein had a tendency to dimerize in the absence of vesicles?
So in case I didn't make that clear, all the in vitro studies were in the absence of vesicles. It's really remarkable. So the specific activity when you have the juxtamembrane segment goes up by a factor of 70 against the peptide substrate. So that's, of course, suggested by Carpenter's work, but what we've shown is that not only in the absence of vesicles, which would concentrate them, so it's purely in solution, it goes up by a factor of 70, the KCAT over K1. Plus, if you measure the oligomerization state, it switches from monomer to dimer by light scattering, which is at high concentration. But if you now measure concentration dependence going down as low as we can, we don't see a diminution in that specific activity, suggesting that this is probably a dimer that has a KD sub-micromolar, 100 nanomolar, 10 nanomolar. Yes?
What do you think is the mechanism of transfer of this signal from the cytoplasm to the membrane?
Oh, I think there is a very simple model we'll propose, which is floating in the literature anyway, but that just says my time is up. That's my time. But if I may have a couple of minutes. Okay. The simple idea would be, and so if you look at the crystal structures that people have done of EGF coordinated receptor, which is by no means anything I've had any contribution to, but if you combine all the structures, it's clear that when EGF binds and you have a dimeric receptor and you can model exactly where the C-terminus would be, it brings the C-terminus above the membrane close together. Right? So a simple model would be that permits, it doesn't have to have an active component, it just permits the interaction that we've been modeling here. Transmembrane segments do their thing and the justamembrane domain does its thing. So then all you need to do to inactivate this is to have any kind of steric bulk in the inactive conformation which prevents the correct orientations of the transmembrane side. Now, of course, nature being what it is, undoubtedly has layer on layer on top of this, but that's the simplest model I propose.
I think Diego from Mark Lemon's lab is here as well. I don't know Diego. Okay, so he's going to be talking later, so maybe we'll hear more about this. Yeah.
You mentioned this track where once it was the C-terminus running along, and once it was the N-terminus. Is there any sequence similarity between the part, the N
Yes.
terminus?
Yes, there is sequence similarity, and the thing we find most remarkable that for reasons of time I didn't go into is that when EGF receptor is activated, it upregulates the transcription of a protein called MIG6, which shuts down EGF receptor signal. And we had crystallized a portion of MIG6 with the EGF receptor, and for various reasons, the only portion we saw was one that actually sits between the activator and the receiver. So MIG6 just binds there. But now, in hindsight, we look at it, and just upstream of MIG6, the portion of MIG6 that we crystallized, is a virtual sequence identity to our justamembrane match. Almost identical sequence over the justamembrane segment. So what MIG6 is also doing is coming in, binding to the base of the kinase domain, and it's taking out the justamembrane segment from the justamembrane match.
So just the kinase domain, without the justamembrane, remember it's like 200 micromolar, which is really very low dimerization of state.
Yes, it's a monomer. It's above 150 micromolar.
And justamembrane, which is you're saying now 100 nanomolar perhaps.
Well, I don't know what it is. It's less than 1 micromolar. Yes.
200, 300 fold improved
Yes.
dimerization. So what I'm trying to understand is that in cell, it's not a dimer. Basically, it's probably stated as a monomer.
No, there's no. It's a dimer. You mean when you just put in jam kinase?
The whole EGF receptor.
No, it's probably a dimer, is what most people would say based on the literature. So why is it an inactive dimer? Yeah. Why is it an inactive dimer? And I would say I don't. I can speculate based on our analysis. And what I would propose is that what's clear from our data is that the helical segment we've modeled as an antiparallel coil is required for stabilizing this dimer. If you delete that, you lose the dimerization. So there must be something, at the very least what we can say is there must be something at the membrane that is preventing that interaction. So let me finish. So it turns out that if you look at the inactive dimer that we have, it's electrostatically polarized. And so we think that it takes an orientation which keeps the N-terminal segments away until the receptor brings them together.
So normally, I don't know EGF at all. Normally, there's a PNA receptor now, but again, oligomerization figures everything. Normally, in sort of resting state, there's something else actually prevents that. My question is whether what is the structure, what you're explaining in the structural way, is it possible that something else actually is not?
Well, it's always possible, but I think our thinking becomes clouded if we always ascribe things to something else. What I will ascribe it to is that we have a proposed model for an inactive dimer, the elements of which are conserved in EGF receptor family members, and allows the receptor to sort of have a resting state, which needs to be broken. Yes?
Just a quick question. So if there is this sort of conserved scaffold, this conserved kind of inactive conformation interaction with the dimer, how do you get specificity for these?
Yeah, that's a great question. That's a question you would ask if you really know the EGF receptor field. So one of the beautiful things about the EGF receptor field is there's a multiplicity of ligands and 4 receptors. And specific ligands will activate specific receptors. So so that's something we've thought about a lot. If an activated receptor can then phosphorylate inactive receptors, which it can undoubtedly according to the literature, then the specificity must include the kinase domain and the transmembrane segment, which are not identical in sequence. Yes.
So, obviously we know that the monomer EGF kinase is not active, and our simulation show that EGF kinase, the core of kinase, is just very, very different from, say, ABA and SARC, in the sense that if you start from active conformation, you never have the helix alpha C stay in active conformation. It very quickly comes out, and we repeated that many times, and it always comes out, and which is very in contrast with the behavior of ABA and SARC. So that's just something very interesting. But here's what I was curious about. So what's the function of the inactive dimer there? Because you just try to not form the active dimer.
Well, I mean, with something as central as the EGF receptor, the moment you're asked what is the function, you really end up in a big state of confusion. So at the very least, it's hiding the AP2, which is the clathrin. So maybe, so one way to answer that, or say to take what you're suggesting, the inactive dimer is not required to maintain the inactive state, which is pretty stable, right? But maybe it's simply sequestering the C-terminal tail, blocking justamembrane latch formation, preventing clathrin coat trafficking. So that's it.
This sort of general comment seems to be quite a fragile system. Many protein kinds here actually activate. But EGF is mechanisms in order to keep kind of really like to be active, and then also try to keep it inactive.
Yeah, but early in the history of EGF receptor, the VERB oncogene was discovered, and it's in a way it's like SARC. You delete the tail of SARC and you activate SARC, and that is the SARC mutation found in certain renal cancers. In VERB, which is an avian viral form, all that's done is you cut off the head, the extracellular domain is unleashed. So yes. So I don't know why that is that way. Nature could be a little cleverer. Yeah. Yeah, I think so. We should file a complaint. Yeah. If I can, I know I should stop.
Yeah, just a quick question. You mentioned that I was reducing residue in the helical model. Do you expect to see a monomer when you use this reducing?
That's a great question. That's an experiment we haven't done. So what you're really suggesting, I should tell you all these kinases are made in baculovirus, and that's not an excuse for the experiment not being done, but it's the reason. Excuses that not all the experiments have done. Yeah.
So if you look at the resting state, you're saying, okay, so dimers exist, but is it an equilibrium in the membrane between monomers and dimers? Is it favored? I mean, it's not a strong favoring for the dimer, I don't think.
No, it's not. The inactive dimer is clearly not strong based on all the data that people have out there. So, and it's not, the inactive dimer we're proposing is not strong either. If you make the kinase domain alone, which should form that inactive dimer solution, it's a monomer.
But in membrane, you would say it should be.
It may be, it'll be stronger, but we don't know.
Okay, so let's take a break.